All right, so we're going to talk a little bit about problems with photosynthesis that plants have and how they can overcome those problems. So what happens is if oxygen is greater than CO2 in the leaves, then rubisco, remember that's the enzyme that binds RUBP to carbon dioxide at the beginning of the Calvin cycle, will fix oxygen instead of carbon dioxide. Rubisco is not a very good enzyme in that sense because its active site isn't specific to just carbon dioxide. It can also use oxygen in its active site. Not a very good one. And so what ends up happening is it produces this weird 5-carbon compound, ends up being produced. And now the cycle can't continue, okay? And that's the key part, is the cycle can't continue. And so now you end up with this weird thing, and it really, the key part of it is it decreases primary productivity. Okay, because instead of producing glucose, nothing's being produced as long as there's more oxygen than carbon dioxide. Okay, now the issue is, and we're going to kind of look at this when we look at transpiration, is when are we going to have more oxygen than carbon dioxide in a cell? And the answer is when we're worried about water loss, because we're going to close the stomata in the leaves. Okay, and so plants have adaptations to fix that. So one is what's called a C4 plant. And a C4 plant, normal plants are called C3 plants, okay? And then a C4 plant is actually just a special plant that fixes carbon dioxide as a 4-carbon sugar. So it turns, as soon as carbon dioxide comes in, it turns it into a 4-carbon sugar, so more carbon dioxide can come in, okay, because it's just moving down a concentration gradient. And what they do is they do separate, this is the key part of C4 plants, is they do separate spaces for Calvin cycle. Okay, and so what's going to happen is in the mesophyll cell, these are the two different cells that are going to be involved, or the two different spaces. Okay, so we have the mesophyll cells. Oops, only 2 L's there, not this third L. And we have a bundle sheath cell. And so what we're going to see is that carbon dioxide is going to come into a mesophyll cell. Okay, here it comes. And it's going to bind with an enzyme called PEP carboxylase. And it's going to turn it into a 4-carbon sugar. Okay. And then, the way it does that is PEP carboxylase uses, you probably guessed it, PEP, to combine with CO2 to form a 4-carbon sugar. And then that 4-carbon sugar will then go into a bundle sheath cell, okay, where at this point it gets split back into carbon dioxide to go into the Calvin cycle. And it also turns into a molecule called a pyruvate, which is a 3-carbon molecule. And then that pyruvate gets turned back into PEP in the mesophyll cell, and then it can combine with another carbon dioxide. The reason why they do this is so that, yeah, you're going to build up a bunch of these 4-carbon molecules, so more carbon dioxide can come in when the stomata are open. As much carbon dioxide as possible can come in because it's going to move down its concentration gradient. If you just had carbon dioxide just sitting around in the mesophyll cells waiting to be used in the Calvin cycle, you might end up not having enough carbon dioxide. So just kind of, and it also kind of stores up a bunch of carbon so that it's available when the Calvin cycle is happening. Okay. A different adaptation for the same problem is what we call a CAM plant. Oh, by the way, sorry, going back to the C4 plants real quick, just some examples of C4 plants. Very common examples. Actually, most of the plants that we have that live around us are C4. Corns, sugar cane, stuff that makes sugar, although beets also make sugar, and grasses, okay? And then a lot of the plants that live in our area are also C4 plants, okay? Another example, or another adaptation to recover from the exact same problem is called a CAM plant. And a CAM plant stands for it's the type of acid metabolism. It's called crushulation acid metabolism. Don't worry about it. The key part of this one is that it's in the same structure, but they just do it at different times of the day, okay? So different times. And so here's what happens in this one. We're actually I'm gonna do like a little dotted line. This is the same cell though. This is what's happening in the cell at nighttime. And this is what's happening in the daytime. So at nighttime, the stomata are gonna be open because because they're not really worried about water loss. It's cooler, and so CO2 enters. So here's my little CO2. It's going to go in, and it's going to turn into what we call an organic acid. And we're going to build up a bunch of this acid throughout the night as the CO2 enters, blah, blah, blah. And then now during the day, the stomata are now closed, so we're not going to be bringing in any CO2. So we're going to take this acid and just convert it right back into CO2, which can enter the Calvin cycle. And we can go ahead and produce our sugar, not a problem, all that kind of stuff, okay? So in this case, CO2 is going to be released. So all we're doing is just building up our carbon when the stomata are open so that we have enough to run our Calvin cycle when the stomata are closed and no carbon dioxide can come in, okay? It's just a way to recover from photorespiration.